Welcome to another deep dive. Today, we're tackling a really foundational topic, something that honestly can feel pretty dense sometimes, the regulation of gene expression. We're digging into Chapter 4 of the updated Medical Physiology by Boron and Boulpaep, yeah, that big one. Our goal here is, well, to cut through that jungle of jargon. We want to pull out the really vital stuff, the nuggets of knowledge, and make gene expression not just, understandable, but actually kind of fascinating. Think of this as your shortcut to really getting a grip on this critical chapter, connecting the tiny world of DNA to how your whole body works.
Exactly. We're going to break down how that single set of DNA instructions manages to create all the different kinds of cells you have. You know, how a liver cell makes albumin, but a muscle cell makes myosin, even though they have the same DNA. And crucially, how those instructions adapt to what's going on around you, your environment. We'll start right at the beginning, build it up concept by concept, and show you exactly why this matters for understanding health and, well, disease too.
Right. So if you've ever wondered how your body just knows to make certain proteins only when and where they're needed, or how one single gene can somehow lead to multiple different protein versions, yeah, you're probably in for some major aha moments today. Let's unpack this. Okay, first off, let's set the stage. You've got something like 30,000, maybe 40,000 distinct genes in your genome. But here's the kicker. In any one cell, maybe only 10,000 of those are actually active, being turned into proteins. So what's going on there? Why the huge difference?
Yeah, that really leads us to two fundamental ideas right away. First is tissue-specific gene expression. Like you said, how do cells in different tissues end up looking and working so differently when they have identical DNA? Your liver cells make albumin, muscle cells don't, muscle cells make myosin, liver cells don't. It's all about which genes are switched on or active and which are kept off or silent in each cell type. It's selective reading of the blueprint.
Selective reading, I like that. Okay, what's the second idea?
The second one is inducible gene expression. See, genes aren't just stuck on or off at a constant level. Their expression, it changes. It responds to signals from outside the cell, from the environment. Think about, say, low blood sugar. Your pancreas releases glucagon. That hormone signals liver cells to massively ramp up the expression of an enzyme called PECK, which is crucial for making more glucose. That's dynamic regulation in action, real-time adjustment.
Okay, dynamic and specific. Let's take it back even further, though, the absolute basics, this central dogma of molecular biology. Information flows from DNA to protein. How does that actually happen?
Right, the central dogma. It's fundamentally a two-step process. First step, transcription. Think of DNA as the master blueprint, kept safe inside the nucleus. Transcription is making a working copy of a specific gene from that blueprint. That copy is RNA, made primarily by an enzyme called RNA polymerase II, and this RNA copy is almost identical to one strand of the DNA, except uracil U replaces thymine T.
Okay, so transcription makes the RNA copy. What's next?
Step 2, translation. This is where that RNA copy gets read to actually build the protein. The genetic code in the RNA sequence is deciphered by transfer RNA, or tRNA. Each tRNA brings in the correct amino acid, and they get linked together one by one, forming a polypeptide chain. This happens on ribosomes, which are out in the cytoplasm outside the nucleus. So that messenger RNA of mRNA is the critical go-between. It carries the message from the nuclear blueprint out to the protein factories.
DNA to RNA to protein.
Got it.
Transcription and then translation. Now, what does a gene actually look like on that DNA strand? You said it's not just one continuous code?
Exactly. It's more structured.
Wow.
If you picture a typical eukaryotic gene, it's a segment of DNA that gets transcribed. It starts at a specific point, the transcription initiation site, sometimes called the cap site,
Yeah.
and runs all the way to a transcription termination site. But importantly, flanking this transcribed region, both upstream, the 5-foot end, and downstream, the 3-minute end, you have these 5-foot and 3-minute flanking regions. These don't get transcribed into RNA, but they contain absolutely crucial DNA sequences called regulatory elements. These control the whole process.
Ah, okay, so the control switches are built into the DNA nearby.
Precisely. Now, the RNA that's initially copied,
Yeah.
it's called the primary transcript, or sometimes heterogeneous nuclear RNA, hnRNA, like the raw footage.
Raw footage. How so?
Because most genes in eukaryotes, like us, contain alternating sequences. You have exons, which are the segments that actually contain the code for the final protein, and will be present in the mature mRNA. And these are interrupted by introns, which are non-coding sequences that aren't present in the final mRNA. The primary transcript contains both the exons and the introns. It's all mixed together initially.
Okay, so raw footage with extra bits, the introns. How do we get from that to the final cut, the functional mRNA ready for translation?
Right, it needs processing. And this involves 4 key steps, all happening inside the nucleus. First, pre-mRNA splicing. This is the editing process. The introns are precisely snipped out, and the exons are stitched together. This makes the mature mRNA shorter and, interestingly, not perfectly lined up with the original DNA coding strand anymore because the introns are gone.
Snipping and stitching. Okay, what else?
Second, a special cap gets added to the very beginning, the 5' end. It's called a 5-methyl cap. It's an unusual chemical structure, and it's absolutely essential for the mRNA to be exported from the nucleus and recognized by the ribosome for translation.
Like a ticket out and an ID badge. Got it.
Third, the RNA transcript gets cut, or cleaved, near its 3' end. This happens about 20 nucleotides downstream from a specific signal sequence, often called the polyadenylation signal. And fourth, right at that cleavage site, a long string of adenine bases is added, maybe 100 to 200 of them. This forms the poly-A tail, and this tail really helps stabilize the mRNA, protecting it from getting degraded too quickly.
So splicing, capping, cleavage, and adding a tail. 4 steps to get a mature mRNA. And once it's ready, how does the rhizome actually read it? Where does it start and stop making the protein?
Good question. The mature mRNA has a central coding region. This is what's actually translated into the amino acid sequence of the protein. This coding region is flanked by a 5' untranslated region, UTR, and a 3' untranslated region, UTR. Translation always starts at a specific 3-letter code, the start codon, which is AUG. This also codes for the amino acid methionine. The ribosome then moves along the mRNA, reading the codons, until it hits one of 3 specific stop codons, UAG, UAA, or UGA. That signals the end, and the protein gets built from its N-terminus, corresponding to the 5' end of the coding region, to its C-terminus, corresponding to the 3 codon.
Okay, that makes sense. From AUG to a stop codon. Now, you mentioned the DNA blueprint is in the nucleus, but DNA is incredibly long, like a meter long. How does all of that fit inside a tiny nucleus just micrometers across? It can't just be floating around loosely.
Absolutely not. It's an amazing packaging feat. About a meter of DNA has to cram into a space maybe 10 micrometers wide. So DNA is meticulously packaged into this higher order structure we call chromatin. The basic unit, the building block of chromatin, is the nucleosome.
Nucleosome.
You can picture it like this. About 147 base pairs of DNA wrap around a core complex of 8 proteins. These are called histone proteins. It's like DNA winding around a spool. 2 full turns around this histone octamer, made of H2A, H2B, H3, and H4.
Beads on a string, right? I remember that analogy.
Exactly. The nucleosomes are the beads, the DNA is the string, and there's another histone, H1, that links things together. This beads on a string structure is 11 nanometer fiber, and then it gets further coiled and folded into a thicker 30 nanometer fiber and even more condensed beyond that.
Wow. So if it's packed that tightly, how does the cell actually get to a specific gene to transcribe it? It seems like it would be completely inaccessible.
That's the crucial next question. For transcription to happen, this tightly packed chromatin structure must be loosened up, at least temporarily, in the region of the gene. This process is called chromatin remodeling.
Remodeling, so changing the structure, how does that work?
One key mechanism is histone acetylation. Remember those histone proteins forming the core? They have tails sticking out, and these tails have amino acids called lysines, which are positively charged. This positive charge lets them bind really tightly to the negatively charged DNA backbone. Think of it as clamping the DNA down, keeping genes inactive.
Okay, positive histones grabbing negative DNA.
Exactly. Now, enzymes called histone acetyltransferases, or HATs, come along and add acetyl groups to those lysines. This neutralizes the positive charge. It weakens the interaction between the histones and the DNA. It effectively loosens the grip, making the DNA more accessible to the transcription machinery, the proteins needed to start copying the gene.
So adding acetyl groups loosens it up. Is there a way to tighten it back down?
Yes, absolutely. Other enzymes called histone deacetylases, or HEACs, do the reverse. They remove the acetyl groups. This restores the positive charge, tightens the histone DNA interaction, and generally inhibits transcription. It's a dynamic balance.
Acetylation on, decetylation off, basically. Are there other remodeling methods?
Yes. Another important way involves large protein complexes, like the Swiss NF family. These complexes use the energy from ATP to literally slide or partially eject nucleosomes, physically pushing the DNA away from the histones, making it accessible.
Okay, so multiple ways to open up the chromatin. It really sounds like gene expression isn't just one event, but a whole series of steps. Where can the cell actually exert control along this path from DNA to protein?
You're right. It's not a single switch. It's a multi-stage process, and the cell can regulate it at pretty much every stage. We can think of about 8 main steps. 1, chromatin remodeling, which we just talked about, making the DNA accessible. 2, initiation of transcription. This is huge, getting RNA polymerase recruited to the gene's promoter and starting the RNA synthesis, often the most critical rate-limiting step in eukaryotes.
Okay, getting it started is key. What else?
3. Transcript elongation, controlling how efficiently RNA polymerase moves along the DNA. 4. Termination of transcription, making sure it stops at the right place. 5. RNA processing. All that splicing, capping, and tailing, we discussed lots of potential for regulation there.
Right, like choosing which exons to include.
Exactly. We'll come back to that. 6. Nucleocytoplasmic transport, controlling the export of mature mRNA from the nucleus. It's a quality control checkpoint too. 7. Translation, actually synthesizing the protein on the ribosome. Control can happen here. And 8. mRNA degradation, controlling how long the mRNA message lasts in the cytoplasm before it's broken down.
Wow, 8 different places. So while starting transcription might be the most common control point.
It is, but cells absolutely modulate all of these steps. Think about DNA methylation, another chemical tag on DNA itself that can silence genes globally, or alternative splicing generating different proteins from one gene. mRNA stability is also a huge factor in determining how much protein you actually get.
Okay, let's focus on that key step then, initiating transcription. What actually does the regulating here? We mentioned regulatory elements on the DNA. What binds to them?
The main players are proteins called transcription factors. These are proteins that bind to specific DNA sequences, those regulatory elements, and then they either stimulate or inhibit the transcription of that gene.
Okay, transcription factors bind DNA.
Right, and we can think of them as transacting factors because the proteins themselves are made from genes located elsewhere and they travel to the gene they regulate. The DNA sequences they bind to, the regulatory elements, are cis-acting factors because they on the same piece of DNA as the gene they control. Cis is meaning on this side.
Transacting proteins, cis-acting DNA elements. Got it.
Now, for most protein-coding genes, the enzyme doing the actual RNA synthesis is RNA polymerase II or POL2. But POL2 can't just find the start site and go on its own. It needs a whole crew of other proteins called general transcription factors, things like TFIA, TFIAB, TFIA, and so on. Together, POL2 and these general factors form what's called the basal transcriptional machinery or the pre-initiation complex. This whole complex assembles at a specific region on the DNA right near the start of the gene, called the gene promoter.
The promoter, so that's the landing pad for all this machinery.
Exactly. The promoter is a crucial cis-acting regulatory element. It determines where transcription starts and which direction it goes. Promoters vary a bit, but a common feature in many is the TATA box. It's a specific DNA sequence, usually about 30 base pairs upstream from the actual start site. The general transcription factor, TFID, recognizes and binds to the TATA box, which helps position everything correctly.
But you said not all genes have a TATA box.
That's right. Many genes, especially housekeeping genes, that are always on at low levels are TATA-less. They use other DNA sequences called initiator elements right at the start site to position the machinery. And close to the promoter, within maybe 100 base pairs, you often find other short DNA elements, like the GC box and the CTATE box. These are binding sites for other transcription factors that help recruit the basal machinery more efficiently.
Okay, so the promoter gets the basic machinery in place. But what about really cranking up expression or turning it way down? You mentioned elements far away.
Right. That's where enhancers and silencers come in. These are other cis-acting DNA elements, but they can be located hundreds, even thousands of base pairs away from the promoter. Enhancers are binding sites for activator proteins, transcription factors that boost transcription levels, sometimes dramatically. Silencers, also called negative regulatory elements or NREs, are binding sites for repressor proteins that inhibit transcription.
And they can work from really far away, and even if flipped around, how?
It's pretty amazing. The prevailing model is the DNA looping model. It proposes that the DNA physically loops out, bending around, so that the enhancer or silencer element, with its bound transcription factor, can come into direct physical contact with the basal transcriptional machinery assembled at the promoter. Imagine bending a flexible rod to bring two distant points together. This interaction then influences the rate of transcription initiation.
DNA looping. That's a cool visual, bringing distant regulators right to the starting gate.
Exactly. And sometimes this regulation isn't just gene by gene. Sometimes entire chromosomal regions containing multiple related genes are controlled as a unit.
Ah, like the globin genes you mentioned.
Precisely. That's where locus control regions, or LCRs, come in. These are super powerful cis-acting elements that orchestrate the expression of a whole cluster of genes within a chromosomal domain. The beta-globin LCR is the classic example. It's located far upstream, but it's absolutely essential for the high-level, tissue-specific, and developmentally timed expression of all 5 globin genes in that cluster.
And the clinical relevance is pretty direct there, right?
Absolutely. Certain types of beta thalassemia, a serious anemia, are caused not by mutations in the globin genes themselves, but by deletions that remove parts of the LCR. Even with perfectly normal globin genes, if the LCR is damaged, they just don't get expressed properly. It underscores how crucial these long-range regulators are.
Wow. Okay, so these transcription factors are the real conductors of the orchestra. What are they actually like? How do they work at the molecular level?
They are truly remarkable proteins. One key feature is their modular construction. They typically have distinct, separable domains or parts. There's usually a DNA binding domain that recognizes and latches onto its specific DNA sequence. And then there's a transactivation domain, or repression domain, that interacts with other proteins like the basal machinery or coactivators to actually influence transcription.
Modular, like building blocks, you can swap parts?
Pretty much. You can often experimentally swap the DNA binding domain of one factor with the activation domain of another and create a functional hybrid protein. It highlights their independent functions.
How do they recognize specific DNA sequences so well?
It comes down to chemistry. The DNA binding domain makes specific non-covalent contacts, mostly hydrogen bonds, with the edges of the DNA bases exposed in the major and minor grooves of the double helix. Each factor recognizes a sort of consensus sequence, a family of closely related sequences, rather than one single perfect match.
And you said they often work together.
Yes, very often. Because any given short DNA sequence might occur randomly quite often in the genome, specificity and high-level expression usually require multiple transcription factors binding to multiple regulatory elements near a gene. This leads to synergy, where the combined effect of multiple factors is much greater than just adding up their individual effects. They might help each other bind to DNA, or they might cooperatively interact with the basal machinery.
And they often work in pairs, dimers.
Correct. Many transcription factors, especially families like the BZIP and BHLH factors, function as dimers, either 2 identical subunits, homodimers, or 2 different but related subunits, heterodimers. This dimerization is often essential for proper DNA binding and can fine-tune their activity and specificity. For instance, the MyCIC protein is a much stronger activator when paired with its partner, MAX.
Are there different structural types of these DNA binding domains, different ways they grab the DNA?
Yes, they're grouped into families based on their 3D structure and how they interact with DNA. Think of them as different kinds of molecular hands or grips. One common type is the zinc finger. Here, a loop of the protein is stabilized by a zinc ion, forming a finger that pokes into the DNA's major groove, making specific contacts. Steroid hormone receptors use these.
Okay, zinc fingers. What else?
Then you have the basic zipper, BZIP family. They bind as dimers. Each half has a basic region rich in positive amino acids that contacts the DNA, and a leucine zipper region that, well, zips the 2 halves together through hydrophobic interactions.
Like molecular tweezers or scissors.
That's a decent analogy, yeah. And similar are the basic helix, loop helix, BHLH factors. They also bind as dimers, using a basic region for DNA contact and helix, loop helix structure for dimerization. MyoD, crucial for muscle development, is one of these. And there's the helix turn helix, HTH motif, found in homeodomain proteins, important development. It uses one specific alpha helix to recognize the DNA sequence.
So different structural solutions for binding DNA. Now, you also mentioned coactivators and copressors. They don't bind DNA themselves.
Exactly. Coactivators are helper proteins. They interact with the DNA-bound activators, but don't bind DNA directly. They often act as bridges or adapters, linking the activator, maybe bound way out at an enhancer, to the basal transcriptional machinery sitting at the promoter. A key example is CBPP300. It not only acts as a bridge, but also has HAT activity itself, contributing directly to chromatin remodeling. Copressors play the analogous role for repressors, helping them to shut down transcription, sometimes by recruiting HGCs.
Okay, so putting it all together, how do activators, maybe with co-activators, actually boost transcription once they're connected to the promoter region?
There are 3 main ways, and they often work together. First, they enhance the recruitment of the basal transcriptional machinery. They make it much more likely that pulled to second and the general factors will assemble correctly at the promoter. That DNA looping is key here. Second, they can trigger chromatin remodeling. Activators can directly recruit HATs or work with coactivators like CBP that have HAT activity. This acytylates histones, loosens chromatin, and makes the promoter more accessible. And third, they can directly stimulate the activity of RNA polymerase II itself, perhaps by influencing modifications on POL2 that are needed for it to start elongating the RNA chain effectively.
Recruitment, remodeling, and direct stimulation. Makes sense. So if activators are the gas pedal, what about the brakes? How do repressors turn genes off?
Repressors are just as crucial. They ensure genes are off when they shouldn't be on vital, for tissue specificity and for turning off responses quickly. They work in roughly 3 ways. 1. Competition. Simple enough. The repressor binds to a DNA site that overlaps with an activator site, physically blocking the activator from binding, like putting a cap over the activator's parking spot.
Okay, just blocking access.
2. Quenching. Here, the repressor doesn't necessarily block the activator from binding DNA, but it binds directly to the activator protein and masks its activation domain. It essentially silences the bound activator, or might prevent the activator from even entering the nucleus.
Interfering directly with the activator protein.
Right. And third, active repression. These repressors bind to specific silencer elements on the DNA and have dedicated repression domains. These domains can directly interfere with the basal transcriptional machinery, or, very importantly, they can recruit core pressors. And some core pressors, like NCOR or SMRT, are very effective because they recruit HDACs, those histone deacylases. This removes acetyl groups, compacts the chromatin, and really shuts the gene down hard.
So repressors can block, quench, or actively shut down using HDACs. That's a lot of control. Now, the cell makes these transcription factors, but can it also tweak their activity after they're made? Like a faster way to respond than making whole new proteins?
Absolutely. This is post-translational modification, and it's a huge layer of rapid control. The most studied is probably phosphorylation adding phosphate groups. This can dramatically change a factor's behavior. It can affect whether it can get into the nucleus, how tightly it binds DNA, or how well it activates transcription.
So phosphorylation can turn them on or off or move them around.
Exactly. Think of NF key in the immune system. It's held inactive in the cytoplasm by a protein called IB. When the cell gets a signal, IB gets phosphorylated, which targets it for destruction, freeing NFT to rush into the nucleus and turn on inflammatory genes.
Wow, quick release mechanism. What else besides phosphorylation?
Another cool one is site-specific proteolysis. Some factors are made as inactive precursors and need to be cut at a specific spot to become active. A great example is SREBP, involving cholesterol metabolism. It sits in a membrane. When cholesterol is low, enzymes cleave it, releasing a fragment that goes to the nucleus to activate genes for cholesterol synthesis and uptake.
Activated by cutting. Clever.
And there's a whole host of other modifications too. Acetylation, methylation, glycosylation, adding sugars, even adding small proteins like ubiquitin or SC2O. These can affect stability, like polyubiquitination often targets a protein for degradation, but monoubiquitination can actually activate some factors. They can affect localization, dimerization, DNA binding, coactivator interaction. It's an incredibly complex fine-tuning.
Okay, so the factors themselves can be modified. How do signals from outside the cell hormones, growth factors, actually trigger these changes and alter gene expression way down in the nucleus?
That's the job of signal transduction pathways. These are molecular relay races that carry a signal from the cell surface to the nucleus, often culminating in the modification of transcription factors. Let's take the CAMP-dependent pathway. A signal outside raises intracellular CAMP levels. CAMP activates protein kinase A, pKa. pKa then travels into the nucleus and phosphorylates a transcription factor called CEB. This phosphorylation dramatically increases CEB's ability to bind its coactivator, CBP. CBP then helps recruit the basal machinery and also has HAT activity remodeling chromatin, boom gene activation, triggered by the initial signal.
A direct line from signal to pKa to CEB to gene activation.
Or consider the RAS-dependent pathway. Many growth factors activate receptor tyrosine kinases, RTKs, on the cell surface. This triggers a cascade involving RAS, a small signaling protein, ultimately activating MAP kinase, MAPK. MPK then goes into the nucleus and phosphorylates factors like c-MYC, c-gene, c-Phos, key regulators of cell growth and proliferation.
So growth factor signal leads to phosphorylated MYC, Jun, Phos.
Exactly. Another pathway is the JAK-STAT pathway, used by many cytokines and hormones. Receptors activate associated JAK kinases. JAKs phosphorylate cytoplasmic proteins called STATs. Phosphorylated stats form dimers, translocate to the nucleus, bind DNA, and directly activate target genes. Very direct route.
Signal to J to stat to DNA.
Okay, and then there are the nuclear receptors. These are really cool because the receptor is the transcription factor. Lipid-soluble signals like steroid hormones, glucocorticoids, estrogen, thyroid hormone, vitamin D, retinoic acid, they can diffuse right through the cell membrane. Inside, they bind to their specific nuclear receptor protein. This binding event transforms the receptor into an active transcription factor.
So the hormone itself activates the transcription factor directly.
Pretty much. Some receptors, like the glucocorticoid receptor, are waiting in the cytoplasm bound to chaperone proteins like HSP90. Hormone binding releases them, they dimerize, enter the nucleus, and bind to specific DNA sequences called hormone response elements, HREs. Others, like the thyroid hormone receptor, might already be in the nucleus, sometimes even bound to DNA, but binding the hormone is required for full activation, often by swapping copressors for coactivators. They then activate transcription using all the mechanisms we discussed, recruiting basal machinery, coactivators with HET activity, chromatin remodelers.
And these nuclear receptors often work as pairs too, right? Heterodimers?
Yes, many important ones like the receptors for vitamin D, VDR, thyroid hormone, TR, and retinoic acid, RAR, preferentially bind DNA as heterodimers with another nuclear receptor called RXR, retinoid X receptor. What's fascinating is that the spacing between the DNA half sites they bind to can determine which heterodimer binds best. There's even a 3-4-5 rule describing the optimal spacing for VDR-XR, TRXR, and RAXR pairs, respectively.
Wow, specific spacing rules. Is there clinical relevance here?
Huge relevance. A classic example is acute promyelocytic leukemia, APL. In many APL patients, there's a chromosomal translocation that fuses the PML gene with the retinoic acid receptor alpha gene, RIA. This abnormal PML-RIA fusion protein acts as a potent repressor. It binds copressors much more tightly than normal RI, even at normal physiological levels of retinoic acid. This blocks the differentiation of myeloid precursor cells, leading to leukemia.
So the fusion protein keeps differentiation genes turned off.
Exactly. But here's the amazing part. Treating these patients with high doses of all-trans-retinoic acid, ATRA, a form of vitamin A, can overcome this. The high dose forces the copressor complex to dissociate from the abnormal PML-RRI protein. This allows differentiation genes to turn on, the leukemic cells mature, and patients often achieve complete remission. It's a landmark example of targeted therapy based on understanding transcription factor function.
That's incredible. Understanding the mechanism led directly to a life-saving treatment.
It really is. And beyond single pathways, you often see coordinate regulation, where one physiological stimulus triggers a transcription factor that then controls a whole battery of genes needed for that response. Think about hypoxia, low oxygen. Under normal oxygen, a transcription factor called HIF1, hypoxia-inducible factor 1 alpha, is constantly being modified by oxygen-sensitive enzymes and targeted for degradation. It's kept at very low levels.
Okay, oxygen keeps HIF1 low.
But when oxygen levels drop, hypoxia, those modifying enzymes become inactive. HIF1 is no longer targeted for degradation. It rapidly accumulates, enters the nucleus, partners with another protein, and activates a whole suite of genes needed to cope with low oxygen. Genes for enzymes involved in glycolysis, making ATP without oxygen. Genes for VEGF, stimulating new blood vessel growth to bring more oxygen. Genes for erythropoietin, boosting red blood cell production. It's a master regulator of the hypoxia response.
One factor coordinating a whole survival program. Amazing. We started way back talking about chromatin structure. How do really long-term changes in that structure, or even marks on the DNA itself, affect gene expression? Things that might even be passed down through cell division.
Right. This brings us into the realm of epigenetic regulation. These are heritable changes in gene function that occur without changing the underlying DNA sequence itself. Remember, chromatin exists in two main forms. Heterochromatin, which is highly condensed, packed tight, and generally transcriptionally silent or inactive, often found in repetitive DNA regions. And euchromatin, which is more open, relaxed, and contains the genes that are actively being transcribed or are poised for transcription.
Closed versus open chromatin.
Exactly. And chemical modifications to the histone proteins are key determinants of these states. We talked about acetylation promoting openness, euchromatin. But histone methylation adding methyl groups to lysines or arginines on histone tails is also incredibly important and complex. Depending on which amino acid gets methylated and how many methyl groups are added, mono, di, or trimethylation, it can signal either activation or repression. For example, methylation of histone H3 at lysine 9, H3K9AE, or lysine 27, H3K27E, is strongly associated with transcriptional repression and heterochromatin formation. These marks recruit specific proteins that help compact the chromatin. This creates a kind of histone code that dictates chromatin structure and gene activity.
A code written in histone modifications leading to long-term silencing.
Yes, these chromatin states can be very stable and maintained through cell division, leading to long-term gene silencing. Classic examples include X inactivation, where female mammals permanently silence one of their two X chromosomes by converting it largely into heterochromatin, or genomic imprinting, where certain genes are expressed only from the copy inherited from the mother or only from the father's copy due to differential epigenetic marks established in the germline. And also just general tissue-specific gene silencing during development genes needed only in neurons gets stably silenced in muscle cells, and vice versa.
So histone mods create stable on-off states. What about marks directly on the DNA?
That's DNA methylation. This involves adding a methyl group directly to cytosine bases, typically where a cytosine is followed by a guanine, a CpG dinucleotide. These CpG sites often cluster together in regions called CpG islands, frequently located near the promoters, start sites, of genes. And generally, high levels of DNA methylation within these CpG islands are strongly correlated with gene inactivation, with gene silencing.
How does DNA methylation silence genes?
It works in a couple of ways. Sometimes the methyl group itself might physically interfere with the binding of an activator transcription factor. But more commonly, methylated DNA acts as a binding platform for specific proteins, like methyl-CP2, methyl-CpG binding protein 2. These proteins then recruit other factors, including HTACs, histone deacetylases, and even histone methyltransferases, which modify the nearby histones to create a repressive chromatin environment. So DNA methylation and repressive histone modifications often work hand in hand to lock genes in an off state.
Wow, a reinforcing loop between DNA methylation and histone marks. Okay, we've spent a lot of time on transcription, which is clearly huge. But what about control after the RNA is made? Post-transcriptional regulation.
Hugely important. Just making the initial RNA transcript is only part of the story. One major mechanism here is alternative splicing. Remember those introns and exons in the primary transcript? The splicing machinery doesn't always have to join the exons together in the same linear order. It can make choices.
Choices like what?
Well, it can skip an exon entirely, a cassette exon, or use different splice sites at the beginning or end of an exon, making it longer or shorter. Sometimes an intron might be retained. Or there might be mutually exclusive exons, where the machinery has to choose one out of a set of two or more possible exons to include. It can even involve using alternative start sites for transcription or alternative polyadenylation sites at the end.
So from one single gene, one primary transcript, you can generate multiple different mature mRNAs.
Exactly. And because these different mRNAs will have different combinations of exons, they can be translated into different protein isoforms, often with distinct functions or properties. It's an incredibly efficient way for the genome to expand its protein coding potential without needing vastly more genes. Think of the atropomyosin gene. It produces different versions in muscle, brain, fibroblasts, all through alternative splicing from that one gene.
That is mind-blowing. One gene, many proteins, amazing flexibility. What about how long an mRNA molecule actually lasts? Does that vary?
Oh, absolutely. mRNA stability is another critical control point. The half-life of different mRNAs can vary enormously, from just a few minutes for some regulatory proteins to many hours or even days for very stable proteins like globin.
And what determines how long it sticks around?
Several factors. We mentioned the 5-foot cap and the poly A tail. They act as protective elements against degradation by enzymes called ribonucleases. Removal of the poly A to annihilation is often the first step towards degradation. But specific regulatory sequences, often located in the 3' untranslated region, 3' UPR, of the mRNA, play a huge role. These sequences can act as binding sites for proteins or even small RNAs that influence the mRNA's lifespan.
Can you give an example?
A classic one is the regulation of the transferrin receptor mRNA, which imports iron into cells. This mRNA has specific stem loop structures in its 3' UTR called iron response elements, IRES. When intracellular iron levels are low, specific proteins called IRE binding proteins, IRBPs, bind to these IRES. This binding stabilizes the mRNA, protecting it from degradation. So more transferrin receptor protein gets made, helping the cell absorb more iron.
Okay, low iron, ameriprotein binds mRNA stable, more receptor. What happens when iron is high?
When iron levels are high, iron binds directly to the IREBP. This causes the IRBP to change shape and fall off the mRNA's IRES. Now, unprotected, the transferrin receptor mRNA is rapidly degraded. Less receptor is made, preventing the cell from taking up too much iron. It's a beautiful feedback loop controlling iron homeostasis via mRNA stability.
That's elegant. Binding proteins controlling mRNA lifespan. Is there anything else major in post-transcriptional control?
Yes, one more really important mechanism, especially prominent in recent research, RNA interference, or RNAi. This involves small RNA molecules, typically about 22 nucleotides long, that can regulate gene expression, usually by silencing it.
Tiny RNAs controlling genes. How?
There are different types, but a key class is small interfering RNAs, siRNAs. These often originate from double-stranded RNA precursors, which get processed by an enzyme called dicer into these short double-stranded siRNAs. One strand of the siRNA then gets loaded into a protein complex called the RNA-induced silencing complex, or RISC. RISC, guided by the siRNA strand, then finds target mRNAs that have a complementary sequence. And depending on the degree of complementarity and the specific proteins in RISC, it can either lead to the target mRNA being cleaved and destroyed.
So chopping up the message.
Right. Or it can simply block the translation of that mRNA into protein without necessarily destroying it. And intriguingly, some small RNAs, perhaps working through related complexes like ARTs, RNA-induced transcriptional silencing, can even guide modifications back on the DNA or histones, promoting heterochromatin formation and silencing the gene at the transcriptional level.
Wow. Small RNAs affecting translation, degradation, and even transcription itself. That's complex.
It's a huge and rapidly evolving field. RNAi pathways, including those involving related microRNAs, mRNAs, are now understood to be fundamental layers of gene regulation, likely crucial for development, differentiation, and cellular defense. Another whole level of control.
Wow. Okay, we've covered a ton today. I mean, from the basic flow of information, DNA to RNA to protein,
Yeah.
to how DNA is packed, unpacked, remodeled, the whole army of transcription factors, activators, repressors, then the processing of RNA, alternative splicing giving us multiple proteins from one gene, and finally controlling mRNA stability and even using tiny RNAs for interference. It's just intricate.
Yeah.
Gene expression is controlled at basically every single step.
It really is. And it has to be this intricate, it's this incredibly precise, multi-layered regulation that allows a single genome to generate the vast complexity of a multicellular organism. It allows cells to specialize, to form tissues and organs. It allows the body to respond dynamically to signals, to food, to stress, to infection, to maintain that delicate internal balance, homeostasis. And understanding these mechanisms, as we saw with APL, isn't just academic. It's absolutely fundamental to understanding health and diagnosing and hopefully treating an enormous range of diseases, cancer, metabolic disorders, developmental problems, you name it.
Absolutely. So thinking back over everything, what stands out to you as maybe the most surprising or, I don't know, elegant part of this whole system?
That's tough. There's so much elegance. But maybe the interplay between the DNA methylation and the histone code, the way these reversible chemical marks on the DNA itself and on its packaging proteins create this layer of memory, this epigenetic information that dictates long-term gene activity and cellular identity and how it can be stably passed down. It really highlights how structure and chemical modification dictate function at such a fundamental level. It's beautiful, really. What about you?
For me, I think I keep coming back to alternative splicing. Just the sheer efficiency of it. That one gene isn't just one instruction, it's potentially a whole set of instructions depending on how you read it, how you splice it. Like the genome has this built-in capacity for functional innovation, for generating diversity without needing endless genes. That choose your own adventure for making proteins is just mind-blowing efficiency. Well, we really hope this deep dive into gene expression has helped clarify things, maybe connect some dots for you, and maybe boosted your confidence a bit with tackling this complex but absolutely crucial material from Boron and Boulpaep. Remember, you're building that strong foundation, one concept, one pathway at a time.
Absolutely. You are part of the Last Minute Lecture family, and you are definitely capable of mastering this stuff. Don't get discouraged by the complexity. Keep exploring it. Keep asking questions. Keep connecting it back to the bigger picture of physiology. Keep learning.
Couldn't agree more. Until next time, keep diving deep.